So as I mentioned before, we will have time later on for questions, so we can move directly to the next presentation that will be delivered by Dr. Caroline Gorvin. She is a Sir Henry Dale Fellow at the University of Birmingham in UK. She obtained a PhD in 2012 from the University of Oxford, where she was researching on cellular mechanism by which mutations in a chloride proton antiporter cause a renal disorder called Dent's disease. Then Caroline continued in Oxford, undertaking a postdoctoral research investigation on the signaling and trafficking of the G protein coupled receptor, calcium sensing receptor, and the role in calcium homeostasis. Caroline moved to Birmingham in 2018 to establish her own research group, and she is currently researching on the fundamental mechanism by which metabolic receptors assemble in membrane, traffic around the cells, and regulate spatiotemporal signaling. Her group uses dynamic signaling tools and advanced imaging to investigate how human diseases affect this mechanism with the aim to improve treatments of metabolic conditions such as obesity. For all this reason, Caroline is here to talk about calcium sensing receptor signaling and trafficking to provide insights into calcium homeostasis. So if you are ready, Caroline, the floor is yours.
Okay, thank you for the introduction, and I hope everyone can see the slides. So yeah, I'm Caroline Gorvin from the University of Birmingham in the UK. I'm going to be talking about some of the work I did in Oxford, and then I've continued to do in Birmingham, looking at calcium sensing receptor signaling and trafficking to understand calcium homeostasis. So we've already had a great introduction to how parathyroid hormone is secreted, so I'm not going to go into any details of this. Instead, I'm going to focus on calcium sensing receptor, which is expressed at the surface of chief cells of parathyroid glands. And the calcium sensing receptor is a G protein coupled receptor, or a GPCR. It is of the class C type of GPCR. This basically means it has this structure in which we have this large extracellular domain known as a venous flytrap domain. And it has this name because it has this bilobe structure, and calcium ligand binds within this bilobe structure, and this leads to conformational changes trapping the ligand within the extracellular domain. These conformational changes are then transduced to the 7-transmembrane domain, and this leads to GDP for GTP exchange at the G-alpha subunit, because it's the G-alpha subunit that links to effectors and has the enzymatic activity. So upon binding of ligand, so in most cases this is calcium, but it can bind other types of ligands as well. It can bind things like magnesium and lithium. Upon binding, we get activation of these G-alpha proteins, and G-alpha proteins can fit into 4 different categories. So there's the G12, 13 and GS categories that I'm not showing in this presentation because calcium sensing receptor largely doesn't signal via these pathways. And instead, we're going to be focusing on just 2 types of pathway, the GQ11 that couples to phospholipase C, leading to increases in intracellular calcium and increases in the mitogen activated protein kinase pathway. So this is things like ERK1/2, which people will know from is important in survival and cell proliferation. And the calcium sensing receptor can also signal via GIO, leading to a suppression of cyclic AMP and an increase in MAPK. And most of what we know about the calcium sensing receptor comes from studies of mutations in the receptor. So these fall into 2 categories. So loss of function mutations lead to hypercalcemia, and this tends to be categorized in 2 different types of diseases. So it can, in a very small group of people, cause neonatal severe hyperparathyroidism, or NSHPT. And this is usually due to homozygous mutations It's very rarely due to heterozygous mutations, and this can be lethal if it's not detected early. The vast majority of loss of function mutations lead to familial hypocalciuric hypercalcemia, or FHH. These patients present, as I said, with low urinary calcium, high calcium in the blood, and they have high or inappropriately normal PTH. They are often asymptomatic, the vast majority of cases, although some people present with mild symptoms like fatigue and excessive thirst. And then very, very occasionally, we have people presenting with pancreatitis. These patients have a low urinary calcium creatinine ratio, and as I said, they're largely asymptomatic, whereas gain of function mutations that causes hypocalcemia, which is a subtype of hypocalcemia known as autosomal dominant hypocalcemia. Around 70% of these patients are symptomatic, and this is typical symptoms of hypocalcemia, things like paresthesia and muscle spasms and tetany. And in a very rare group of people with ADH, we also see a Barter's-like syndrome. So although we know that calcium sensory receptor mutations cause the most cases of FHH and ADH, which I'll be focusing on mostly in my talk, we also know in 2013 it was published that FHH and ADH can be genetically heterogeneous. So work from the Thacker and the Manstadt groups showed that mutations in the G-alpha-11 protein, by which the calcium sensory receptor signals, lead to FHH type 2 and ADH type 2. These are largely similar to FHH and ADH due to calcium sensory receptor, except FHH2 tends to have a milder calcium phenotype, and ADH2 is often associated with changes in growth. So we have short stature in around half of the patients that present with ADH2. And then a third type of FHH3 is due to mutations in the adaptor protein 2 sigma subunit encoded by AP2S1. And this protein is part of the adaptor protein complex that's important in internalization of the calcium sensory receptor, so movement away from the cell surface to the endosomal pathway. And these patients are often symptomatic, so around 20% of them are symptomatic compared to calcium sensory receptor mutations that are very rarely symptomatic. We often see a greater serum calcium and magnesium in these patients, and because they have a greater serum calcium, they can often overlap more with primary hyperparathyroidism, so it's important to distinguish between the two to avoid unnecessary surgery. So the mutations that we see in calcium sensory receptor, we've seen more than 300 different germline mutations so far that have been reported. If you go on to things like NOMAD, you'll see that there's many more mutations that occur in the calcium sensory receptor, and they may or may not affect function. On this diagram, just showing the extracellular domain, and just to orient you, you can see that the lobe 1 and lobe 2 of the venous flytrap, and the cysteine-rich domain that connects the extracellular domain to the transmembrane domain that's not shown in this diagram. We can see loss of function mutations in blue and gain of function mutations in red. And we know that and although these seem to be spread quite randomly throughout the structure, in fact, we can see some clustering. So for example, around the calcium binding sites, which are shown in purple here, you can see clustering of mutations. We also see a clustering along the dimer interface where the two calcium sensory receptors connect together. And then there's also a clustering at transmembrane 6 and 7, which we know is important in calcium sensory receptor activation. And some residues are the site of loss and gain of function mutations. And some of these, a subset of these represent switch residues. And we've called them switch residues because we think they're important in switching between the inactive and active state, and therefore mutations in these residues. Mutations to one amino acid could cause FHH, and a mutation in the same residue can cause ADH. So in my talk, I'm going to be talking about several different mutations in the calcium sensory receptor and within its pathway. And firstly, I'm going to focus on calcium sensory receptor, 2 different calcium sensory receptor mutations leading to ADH and bias signaling. So what I mean by bias signaling is that if we envisage that we have a receptor signaling via two different pathways, we could think that these were signaling equally by these two pathways. But it's known in GPCR biology that some mutations in receptors and also some ligands, when they bind to receptors, can cause preferential signaling via one pathway over another. And this we call bias signaling. So I'm going to talk about one form of bias signaling what we identified in a patient with an arginine 680 glycine variant. So this was a patient that presented in Denmark, and we worked with collaborators to try and work out how this mutation was causing ADH in this patient. So the arginine 680 residue is located at the top of transmembrane 3, which we know is important in linking across the GPCR to allow G proteins and beta arrestin to bind to receptors. So when we find calcium sensory receptor mutations, we usually do intracellular calcium signaling because this is the first characterized pathway by which the receptor was known to signal. So we, in all of these, throughout this talk, I'm going to be talking about expressing plasmids in HEC293 cells. I know HEC293 cells are not the greatest model, but we don't currently have any parathyroid cell models that that everyone has access to and that can consistently measure calcium sensor receptor signaling. And therefore, we use HEC293s because they don't express calcium sensor receptor, and we can then look at how these mutations are responding in isolation. So we express in our cells either the wild type receptor, our Gly680 mutation that we found in our ADH1 patient, and then a gain of function known mutation in phenylalanine 173. And we could see if you expose the cells to increasing doses of extracellular calcium, we get this nice dose-dependent response, and that we get this leftward shift in our positive control, which we would expect for ADH1, and that we don't see a shift in our patient mutation. So we could conclude from this that it's not affecting intracellular calcium. However, when we looked at phospho-ERK signaling, so this is a type of MAPK signaling that can, as I said, occur downstream of two different types of G proteins, we saw an elevation in both our positive control ADH mutant and also our new mutant. And therefore, what we've shown in this is that this ADH1 associated mutation has normal intracellular calcium, but increases ERK1 too. And this is an important learning point because the vast majority of people only look at intracellular calcium signaling, and we could have concluded from this that this mutation was benign and wasn't causing the phenotype. So it's important to try and look at a couple of different signaling pathways if possible. So we've shown that we have increased MAPK, but we don't have an increase in intracellular calcium. And as I said at the start, MAPK can occur downstream of multiple G proteins. So we wanted to try and determine which of these G proteins was important in signaling, because this might be important for treatments when we develop treatments targeting individual pathways. So we wanted to look at a single assay. So we used a serum response element luciferase reporter assay. So so this is a luciferase reporter that can measure signaling downstream of calcium sensory receptor. So when we have activation of the receptor, we get increases in MAPK, which leads to increases in ERK effectors, and these combine to these serum response elements and produce luciferase, which we can measure on a plate reader. And we performed assays of our mutations alongside our wild type, and then inhibited with a GQ11 inhibitor initially to see whether GQ11 was important in this signaling. And what we could see was that GQ11 inhibitor reduced signaling in our wild type and our positive control mutant, but it didn't reduce signaling in our R680G mutant, indicating that the GQ11 pathway is not important in this increased signaling. So we then moved on to look at the other G proteins, so GIO. So we used pertussis toxins to suppress this signaling pathway, and although we saw a small reduction in our ADH1 mutant, this was still significantly higher than our wild type. So we think that it's unlikely that GIO is important in this signaling. So this then led us to think that it could involve a different pathway. And in a subset of GPCRs, we know that beta-arrestin can signal to MAPK. So we suppressed beta-arrestin 1,2 with siRNAs. I'm just showing you the data from the beta-arrestin 2 siRNA, and we can see that our R680 is suppressed below the wild type level in this. And therefore, we think that this ADH1 mutant is activating a non-canonical beta-arrestin pathway. So we then turned to homology models. So this was done several years ago now. So we use a homology model based on a mGluR structure. Now we have lots of calcium sensing receptor structures from both crystal structures and cryo-EM structures. And therefore, we could have done this much quicker with these newer models. But we could see that in this homology model that the arginine 680 is located at the top of TM3. And when we look closer, we can see that it makes these two connections, these two predicted connections. So either with glutamic acid 767 or with 837, or it could bind to both. So we decided to mutate each of these residues to an arginine, which would break this salt bridge and then test it in our SRE assays. And when we performed this, we could see that the arginine 767 mutant had a similar phenotype to our ADH1 mutant. And therefore, this suggests to us that it's this arginine 680 glutamic acid 767 salt bridge that forms, and we hypothesize that mutation of this residue disrupts the salt bridge, allowing beta-arrestin to bind more readily at the intracellular region, because we know that TM3 and TM6 are important in opening this cleft at the intracellular region, and therefore we think beta-arrestin binds more readily here. So this is a unique non-canonical beta-arrestin pathway that we've shown by studying this mutation. So for our second biosignaling story, we're going to look at another ADH mutation. This is brand new work. This work is currently in revision. And I thought I'd present this because we have a slightly strange phenotype that we've never seen before. So this is a case of ADH1 in which the patient presented with short stature. There's only one report of short stature with calcium sensitive receptor mutation, and when this has been expressed in cell models, this doesn't seem to be, it seems to be a SNP, so it seems like this is just an incidental mutation, whereas in our mutation, as I'll show you, this seems to be a calcium sensitive receptor mutation that is associated with short stature. So short stature is very rare, or doesn't occur in ADH1, but in ADH2, due to GNA11 mutations, it's quite frequent. So we see it in multiple patients and also in mouse models of GNA11 mutations. And therefore, we hypothesize, because of the location of the variant at leucine 723, which is in the intracellular loop 2, because this is located in the G protein binding sites, and because GNA11 activating mutations cause ADH, we hypothesize that this mutation could affect G protein coupling. So we looked at G alpha 11 signaling using Brett assays. So here you can see that we've got, if we expose our cells to increasing extracellular calcium, we've got this nice increase in G alpha activation, and that this elevated in our variant, and that we have a reduction in the EC50. And we could show looking downstream signaling. So rather than just looking at the G protein, if we look at the signaling downstream of this, so IP3 activation that occurs upstream of intracellular calcium, we can see that we've got this dose-dependent increase, and that this is elevated in our arginine 723 mutant. And then if we give a G alpha 11 siRNA, we get this reduction in signaling. So we think this mutation is activating G11. When we looked at other G protein pathways, we could see that they weren't increased by this signaling. So what we've shown is that we've got a bias signaling via G11. And we think that it might be because of this preferential activation of G11 over the other signaling pathways that we're seeing this short stature in our patients. So I've told you two stories about biosignaling. and mutations in the calcium sensory receptor. And now I'm going to go on to the final part of my talk, where I'm going to discuss mutations in the calcium sensory receptor pathway and how they affect spatiotemporal signaling. So as I said at the start of the talk, we have a subset of patients that present with FHH with mutations in the adapted protein 2 sigma subunit. This is usually due to mutations in the arginine 15 residue. And these patients tend to be more symptomatic and have a higher serum calcium than calcium sensory receptor mutation patients. So the AP2 sigma subunit is part of the adapted protein 2, which is a heterotetrameric protein shown as a structure at the top and a cartoon at the bottom. We have this large alpha and beta subunit, which link to clathrin and other accessory proteins, whereas the smaller mu and sigma subunits interact with, so they're able to recognize motifs on membrane proteins to target them from internalization. So they're basically recognizing where receptors are and tells them to internalize. So we hypothesize from this, because we've got mutations in the AP2 sigma subunit, that means that we're probably going to have changes in calcium sensory receptor endocytosis. So we wanted to measure this in our cells, but we knew that we had to take into account that calcium sensory receptor has slightly strange biology because unlike most GPCRs, it's continuously synthesized at the ER, and then this mature calcium sensory receptor exists in pre-plasma membrane vesicles. And it sits in these pre-plasma membrane vesicles until we get changes in extracellular calcium. And then we get this phenomenon known as agonist-driven insertional signaling, where we get a huge increase in calcium sensory receptor exported from these vesicles due to changes in extracellular calcium. And we think that the calcium sensory receptor has developed this mechanism to be able to respond rapidly to very small changes in calcium in the blood. So we knew that if we were looking at endocytosis, we'd also have to take into account this agonist-driven insertional signaling, because we'd be having increased calcium sensory receptor at the membrane, as well as removal from the membrane by endocytosis. So to measure these two components simultaneously, we use this BSEP calcium sensory receptor construct shown here. So this comprises full-length receptor with this N-terminal modification containing a superecliptic fluorine, which is basically a modified GFP that fluoresces maximally at the cell surface. And we could use this to measure total plasma membrane calcium sensory receptor and also record ADIS events. So we can see the calcium sensory receptor sort of popping to the cell surface. And then it also contains this minimal bungarotoxin binding site, which is a 13 amino acid sequence that can bind these fluorescent alpha bungarotoxins that you can buy commercially. So in the basal state, the calcium sensory receptor will be green, and we won't have anything on our alpha bungarotoxin site. But when we add our fluorescent bungarotoxin on the microscope, we'll then be able to see red fluorescence as well. And then when this disappears from the cell surface, we'll know it's because it's bound to a receptor at the cell surface. Because on its own, this dye is cell impermeable. So we recorded these fluorescents using turf microscopy. So turf microscopy is a specialized type of microscopy where you can only see around 100 nanometers at the plasma membrane. So we can only see events that are occurring at the plasma membrane, and we're not obscured by events occurring elsewhere in the cell. And we recorded our cells over 1,500 seconds. We expose our cells initially to basal calcium, and then expose our cells to a massive increase in serum calcium to induce ADIS, and then step back down to basal calcium. In both wild type and our AP2 sigma mutant cells. And this can be seen more clearly if we plot the fluorescence from each of the frames for the whole of the movie. And we can see here in this in basal calcium, we've got very little activity. And when we give this pulse of calcium, we get this massive increase in calcium sensory receptor at the cell surface due to ADIS. And that this appears to be a little bit higher in our AP2 sigma cells. And when we measure by ELISA, we do see an increase in cell surface calcium sensory receptor. And then if we look, we think that this increase in surface expression can be explained partially by looking at the endocytosis, where we can see in our wild type, we get a nice removal from the cell surface, which can be seen quite clearly here when we quantify the fluorescence. Whereas in our AP2 sigma mutant, we've got very little removal from the surface, and we've got very impaired endocytosis. So our mutants have shown that we have increased cell surface expression and impaired internalization. And we think this is because this arginine 15 residue is important in binding to dileucine motifs. So these motifs that are internalization motifs on membranes. And this motif is the one that's been shown in the calcium sensory receptor. We can see in our wild type, we've got a connection between the arginine 15 residue that's mutated in FHH3, which binds to this motif. And then if we mutate this to one of our patient mutations, we can see that we lose this connection. So we think that the AP2 is not able to recognize calcium sensory receptor at the cell surface and not able to internalize it as efficiently. So we have increased calcium sensory receptor at the cell surface, but we can show by signaling assays that we've got a reduction in calcium sensory receptor signaling, which is consistent with FHH. And therefore, that's a little bit confusing, because you'd expect if you've got increased receptor at the cell surface, then you would have increased signaling, because you should just be able to continue signaling forever from the cell surface, as long as we don't have a desensitized receptor. And therefore, we try to think of different mechanisms that could be involved in where we would have an increase in surface expression, but a reduction in signaling, and this reduction in endocytosis. And when we look in the literature from a number of GPCRs, including the PTH receptor, it's now recognized that GPCRs can signal not just from the cell surface, but can signal once they've been internalized. So following internalization, they can go to different locations like the endosome or the Golgi and can spark a second signal. And this can is sometimes known as a sustained signal, and it can occur from multiple sites intracellularly. So we hypothesize that the calcium sensory receptor can signal from the plasma membrane and the endosome. And we assess this using our reporter assays, but instead of giving constant calcium, in this case, we gave a pulse of calcium and then stepped back to basal calcium. And we can see in our vehicle-treated cells that we get this nice increase in activity. And then we see this second wave of activity. And if we expose our cells to DINGO, which is an inhibitor of internalization, we see this first wave of signaling, but we lose this second wave. And therefore, we think this second wave is due to sustained signaling. And therefore, we concluded that AP2 sigma mutations impair calcium sensory receptor sustained signaling. And then finally, in very recent studies, we've been looking at where this signal could be coming from. So we've been using these Brett assays, in which we're looking at recruitment of luciferase tagged G proteins and how they associate with fluorescent markers of different compartments. So at the cell surface, we've been using this KRAS marker. So when GQ is recruited to the calcium sensory receptor, we should get a Brett signal. If it's recruited to the plasma membrane, we'll get a Brett signal because it'll get close to this KRAS. And then upon internalization, if it goes to the early endosome, we'll get this NLUC will suddenly become close to the RAB5 and we'll see a Brett signal. And this can be seen here in these diagrams, where you can see that when we expose the cells to GQ and GI, we get this nice increase in recruitment to the plasma membrane, and that there's less recruitment, but it's still present for GS and G12. And then we use this to see where the calcium sensory receptor and active G proteins were recruited to. So we showed that calcium sensory receptor and GQ goes to RAB5 early endosomes, and the other G proteins don't go to these pathways. And then we assessed multiple different compartments using 12 different markers, and we could see that we get this recruitment to RAB4 positive endosomes, which are important in early endosome to recycled endosome signaling, and RAB9, which is important in late endosome to Golgi signaling. And we think that this might be important in our ADIS signal. And we could show that if we suppress these two RAB proteins, we get this reduction in signaling, and we could show nice co-localization of the receptor with these markers too. So I've shown that calcium sensor receptor activated mutations can stimulate non-canonical signaling pathways to cause ADH1. Calcium sensor receptor may contribute to linear growth and may need to be considered in patients that present with short stature and hypocalcemia. And the calcium sensor receptor can signal by GQ11 from intracellular membranes, and this pathway could be uniquely targeted for neural therapeutics. So for my acknowledgement, so this is basically acknowledging everyone who's contributed to FHH and ADH studies over the last few years. In particular, I should acknowledge that lots of the AP2 work and the first biosignaling study was performed in Raj Thakker's lab, the University of Oxford. And then I continued doing this with my group shown at the top here, using a Sir Henry Dale Fellowship from the Wellcome Trust and Royal Society, where we've continued working on this. But we couldn't do any of this work if we didn't have clinical collaborators who let us know about interesting ADH and FHH mutations. And thank you for listening.
So thank you so much, Caroline, also for this great presentation. I think both presentations were really nice and really help us to have an overview of what is happening with these diseases, trying to understand the molecular or the biological part of the disease. As I said at the beginning, now we have time for questions. So there were a couple of questions already in the chat, but was specifically to congratulate you about the presentation. So well done. I had to say that we had a lot of attendees. So just to let you know that the topic is of great interest. Usually we need some time to start to receive some questions from the audience. So in the meanwhile, I would like to ask to both of you a couple of questions. So you were talking, Caroline, about all the mutations and which could be the impact on the disease. But I would like to know if there is some information of, if you know if there is information, you or Mikkel, about how prevalent are those mutations in the landscape of the disease. So they could represent important proportions of the patients, or is something that is just in the minority of them?
So in terms of you mean how common is FHH and ADH? So they're classified as rare diseases. FHH, it's debated how much we see numbers ranging from around 2 to 3 in 100,000 is what people normally 3%, sorry, is what people normally suggest. But the problem with FHH is that in lots of people it's asymptomatic, and we think that many mutations that are classified in NOMAD are actually mutations rather than just being SNPs as they're seen at the moment. And when we've tested some of these in all 3 of the FHH proteins, they do cause a change in the calcium sensor receptor signaling, and we think that it might be important in regulating the set point.
And are they redundant in the patients? I mean, some patients are harboring several mutations, or they used to be one single mutation for patients?
Yeah, so we see some compound heterozygous mutations in NSHPT, but as I said, they tend to be people who are presenting with severe hyperparathyroidism in infancy. So this is usually, this can be lethal, and They're very, very rare. There's only a very small number of people who've got these mutations. And then, as I said, there's some who are homozygous, but we don't see multiple mutations in very many people.
So there is a question from the chat. It says, "Thank you for the excellent presentation. What is the economic impact of routine genetic testing in FHH?" So I don't know if you, Mikkel, have some insight about that or some guess.
Yeah, so to routinely test in hypercalcemia, was that kind of the question, to do some screening? Well, it's like Caroline says, this is quite a rare disease, and primary hyperparathyroidism is a very common disease. So to think that there is kind of a big gain in testing all these patients, what we of course do is we measure the urine, and if the urinary calcium is very low, we normally first retest, and if it's still low, we can do the genetic testing in these patients. So still, if we do it like this, which is kind of this is the clinical differential diagnostic test, but still, if we do it like this, I think the number is that if the urinary calcium is low, still about 9 out of 10 will still have primary hyperparathyroidism. So it means that to routinely test every patient would be very economically unwise, but genetic testing is quite available. So if you have low calcium values in the urine, and especially if you have a family history, you should definitely consider genetic testing. Wouldn't you agree, Carolyn?
Yep, that's what I would have said, yep.
Okay, so well, one of the ideas of these spotlights is to bring together the clinical and the basic or translational expertise on one topic. So trying to follow with this idea, I would like to ask Mikkel about, if you had to choose, what do you think could be the most relevant clinical need in these diseases, where we could be thinking about the part that the basic translational research could be contributing to address?
So this is, of course, an excellent question, and there are numerous trials on different aspects of calcium disorders. So I think now there is a lot of things happening in terms of treatment of, for example, hypoparathyroidism, and what is the optimal treatment. I think there is problems in kind of, I tried to allude to the fact that still today we are not really concurrent about simple things such as diagnosis, such as what is the which patient to operate in primary hyperparathyroidism, which patients to treat in hypoparathyroidism is still kind of very, it's many unanswered question, and I think that the basal investigations here are actually in all of these themes, they are very valuable.
In the same line, Caroline, do you know if there are some lines of research already addressing how to say correct those mutations in patients where we know what they have already those mutations? I don't know, genetic editing, well, not in the patients obviously, but some experimental approaches to see if we can restore the normal physiology if we correct this mutation. Do you know if there is something on place in this line?
So there isn't any targeted therapies for these particular mutations. Many cases of FHH are asymptomatic, so they don't require treatments. And there are therapies that target the calcium sensory receptor, but these are not targeting specific mutations. In terms of wider bone and calcium disorders, there's obviously there's possibly going to be some need for doing more targeted looking at mutations depending on the disorder, but often these are very rare diseases, so pharma aren't really interested in looking at specific mutations if they're only occurring in very, very few people.
Okay. Okay, so great. That's well. We have one question in the chat. I guess you both can also read it, right? So the question is, what is the cause of the short stature in ADH2 and ADH1 you found with biased signaling? GS11 plays a role in both bone and pituitary growth hormone secretion.
Yeah, so we don't know exactly. So we know that G-alpha-11 is important in signaling downstream of other receptors. So we know it's important in signaling downstream of PTH receptor. It's important in chondrocytes, and it's in differentiation of chondrocytes. And it's thought that in some PTH receptor and G-alpha-S mutations, that we get accelerated chondrocyte differentiation and premature fusion of the growth plate, and that's why we get short stature. That wouldn't explain necessarily how we're getting the effect in calcium sensory receptor, and we don't actually know how it's contributing. We don't know whether G-alpha-11 downstream of calcium sensory receptor has an effect on growth, and really we'd have to develop a mouse model of this specific mutation to be able to look at that in more detail. But I just thought I would report it because it's an interesting finding. It's not really been seen before, so we thought it might be something that we do need to start considering. But at the moment, we've just got this one case, so it might just be that it's contribution, because we know that growth has multiple factors, so it's a polygenic trait. So there could be, when we do GWAS studies, we see hundreds of variants that can contribute to growth. So it could be that calcium sensory receptor is something, and G-alpha-11 is something that contributes, but we don't know the specific mechanism.
Okay, thank you so much. So, well, there is that we have another question in the chat. Is 184 or 134 PTH use in hypopara?
Yeah, so I tried to kind of sort this question a bit out because it's could be confusing, but at the moment there is a drug available at the market, Natpar, which is 184, and this is a longer half-life than PTH134 teriparatide, but it will go out of the market. Takeda has decided that it's not enough in it for them, so they have stopped producing it. So 184 will not be available by the end of 2024. And it also says NY, please. It's okay that I understood this as well, Manuel. Yeah. So it's mainly because that 184 has a much longer half-life, so you can treat it with one injection per day. So if you give 134, it will be the very short half-life of about 1 to 2 hours will kind of not help the patients during the 24 hours. So now it's the new drug in the market now is kind of a mix where you kind of try to increase the half-life with this transcon linker.
Okay, great. So thank you so much. As I said some minutes ago, we had a high number of attendees, so it's meaning that this topic received a lot of interest and attention. So I would like to finish by thanking you for sharing your time and sharing your expertise with us. I hope you enjoy the experience being here in the spotlight, and for sure all the attendees learned a lot from you. So thank you so much. And I would like to finish by inviting you for the next month. In 2 months, in November, we will have the last spotlight of science of this year, and it will be focused on muscle D, metabolic dysfunction associated steatotic liver disease. So I hope you will join us, and wish you the best for these 2 months. Thank you again to the speakers, and thank you to all the attendees. We will see soon. So thank you so much. Bye-bye.